Yes, and thank you for having me today. Of course, I guess I'm sort of having myself. No, this was definitely, this series came together in large collaboration with all of our Walnut colleagues. So huge thank you to all of them for being here and being a part of it and helping plan this all out. So big thank you for that. Yeah, so I just thought that I would cover a handful of diseases that I see in the field every now and then, sometimes more frequent than others, that growers have had questions about. And I figured we'd just do a little bit of ID and a little bit of management, and we'll see how far we get in 15 minutes. Certainly some of the work that you saw today, as well as some of the information in these slides, was produced from research that was funded by the California Walnut Board. So that's your money hard at work. I did want to cover thousand cankers disease because we do see this on occasion and it looks really scary, and it might not be as scary as it looks. So it's good to discuss it and cover it generally speaking. So thousand cankers disease, basically when you're looking at the tree, you'll see these little holes that are D-shaped, and they're about the size of the end of a pencil, a number 2 pencil. And they're usually surrounded by darkening, or we call it necrotic tissue, but dead or dying tissue. So what that is actually the walnut twig beetle. The walnut twig beetle has bored into the tree, and it brought with it some fungi, and that fungi is the Geosmithia morbida fungi. And what's happening is, if the walnut twig beetle was doing its own thing all on its lonesome, it wouldn't be a huge deal. It wouldn't be much of a pest, but unfortunately it brings with it this fungi, which is pathogenic on the walnut tree. And that's where you get that blackening, that darkening, and sometimes almost like an oozy effect there. So current research, this information was just sent to me this weekend from Rick Bostock. So thank you, Rick. And he is present also to respond to any questions if I'm unable to answer them. So the peak walnut twig blight flight period is appearing as though, according to trapping, late May to early June, and often extends into July. Second potential flight period looks as though it might be August through late October. This is again based on trapping studies over 2 sites over 5 years. Research is currently focusing on certain repellents, although no recommendations are available at this time. Initial results indicate that trees with crown gall disease may be at high risk of walnut twig blight attack and the thousand cankers disease. That's the WTB and the TCB there. Other stresses may predispose walnuts to walnut twig beetle attack and thousand cankers disease, but there is no hard data to prove this. But we always want healthy trees, right? Walnut twig beetle can colonize and emerge from crown galls and can carry crown gall bacteria. It is unknown whether Agrobacterium tumefaciens, the causal agent of crown gall, contaminated with walnut twig beetle, can transmit the bacteria to a healthy tree and cause crown gall. So those are all interesting things that are being looked into as we speak. Something that we did see the past few years, like spring, May 2019 and spring, May 2020, we saw what some people were calling it a delayed emergence, but actually, as it turned out, there were just a lot of unfortunately dead branches in the trees. And we later realized that this was related to freeze damage, but this freeze damage was actually occurring in November. This may be related to a dry fall. This may be related to cold enough temperatures, but not necessarily unusually cold, given our experiences. But this is related to the potential fact that temperatures were dropping from very warm temperatures to very cool temperatures in the passage of, say, a day back in November. And unfortunately, the branches didn't have enough time in that 24-hour period of temperature change to change their carbohydrate structure. And so this may very well be related to the fact that we were getting these severe swings in temperature, and the trees were not quite as dormant as they needed to be given those fluctuations in temperature. So it is possible that irrigation after harvest could help. Irrigation water certainly does have a warming effect. You can prune in June. So once you've seen all that damage, you can go back and prune in June, and you can use 50-50 latex paint with water. That's interior latex paint for sunburn protection. Additionally, reducing, like, don't fertilize after August. slow down your irrigation in September. What you're trying to do is get that tree to go into dormancy. You don't want green growth in November. That'll definitely give you freeze problems. Phytophthora, this is what it looks like. This is obviously a severe infection. Hopefully it's not this bad in your orchard, but it can be. This was actually related to floodwaters, like 2 or 3 feet above ground. But this can also happen just from having your irrigation on too long. So avoid 18 to 24 hours of soil saturation. So don't run your irrigation for more than 24 hours at a time. You need good soil drainage to avoid Phytophthora problems. Plant into berms. Don't plant the tree and then form a berm over the crown. Plant into the berm if you have heavy soils. Try to avoid soil compaction. You can use water guards on your sprinklers. Try to promote water infiltration, so cover crops can help with this if you have a heavier area that you need to break up that soil. Additionally, the choice of rootstocks. RX1 has shown a high resistance to a certain species of Phytophthora. Additionally, there is a moderate to high resistance to another species of Phytophthora. So RX1 is a handy rootstock, and that is a UC research-based rootstock release that is available to you, and that rootstock and the work behind it and the future rootstocks, clonal UC rootstocks that are being developed as we speak, are all funded in some part by the California Walnut Board. Chemical control is also an option. You can certainly find this information on UCIPM, and so I'm not going to go deep into it, but those are options for you. You do always want to check with your county to see if there's certain restrictions depending on the time of year. Crown gall, everyone's probably seen crown gall, everyone's experienced it. It can be a real pain in the back end. This is actually Agrobacterium tumefaciens is the bacterial causal agent for crown gall. It's actually genetically modifying the plant, which makes it very difficult to control. That also has that little chip of, genetic modification is actually something that occurs in nature. But with that said, basically what it's doing is it's inserting some DNA into the plant and telling the plant to produce opines and hormones so that the bacterium can feed off of that. Then those opines and hormones look very similar to a compound that tells the plant to produce callus tissue, and you end up with crown gall. This can be very deceiving because it will grow and then it'll stop growing, it'll grow and flake off and grow again. So you get this annual growth, and it may look like it's gone away, but it typically returns. Prevention is paramount. Sanitation is key. Clean your tools, 10% bleach, 70% ethanol. You must clean your grafting tools, your pruning tools. After you've cleaned your pruning tools, make sure that you don't hold the lopper blade ends with your dirty gloves. That will just basically move the bacteria right back onto your pruners. Basically, we're talking about a soil-borne pathogen, and it's in most soils. So if you were digging with your gloves on and now you're doing some pruning and some lopping, you may end up moving it from your gloves to the blades. So just make sure that you handle your loppers properly so that the blades don't touch the ground and your blades don't touch dirty gloves. So pre-plant fumigation is an option. This does work, but this does not work with galls. So it's difficult to penetrate the gall tissue. So you can go out and try to remove as much gall tissue as you can if you're planting walnuts again. Anaerobic soil disinfestation is effective. This is a process in itself that you could spend another 30 minutes talking about, but basically we're talking about using rice bran and water and heat to allow for microorganisms to break down that carbon source, and that effect actually penetrates deeper than fumigation. So research is sort of moving towards, how can we make this affordable and economical and able for growers to adopt. Paradox seedlings are highly susceptible to crown gall. RX1, VX211, and Blatch are not resistant to crown gall, but they are clonally propagated, so that it's a different system that they're produced from. That said, there are ways that nurseries can accommodate that with paradox seedlings. So you just want to be sure that you're talking with your nursery about their practices. With paradox seedlings, they would want to be using tarps for collection, whereas with RX1, VX211, and Blatch, they are not produced. They are not shaken from a tree and collected from the field. They are just cut and clonally propagated in a greenhouse. That said, RX1, VX211, and Blatch still require tool sanitation. And RX1, VX211 are UC releases, which again are from the Walnut Improvement Program, which is where some of that money, basically, if you buy an RX1 rootstock or a VX211 rootstock, you will pay a little bit extra for that. But that's because that cost, that small amount of money, will go right back into producing future varieties and rootstocks for walnuts. Just want to mention Blatch is not a UC release, so we can claim none of that credit. It's just a clonal rootstock that's available. UC is field testing crown gall resistant rootstocks. I actually have one under my wing as we speak. So in the future, there may very well be crown gall resistant rootstocks available. That's still at least probably a 10 to 20 year field trial. So give us a little time. Walnut blackline, this is actually caused by a virus. It's the cherry leaf roll virus. This is a result of infected pollen. So you could almost consider it a sexually transmitted disease for walnuts. So cherry leaf roll virus will spread via infected pollen from an infected tree. It then infects the healthy female bloom. The virus enters the tree. The English scion, so the tasty nut that we like to eat, that English plant on top is tolerant, so it shows no symptoms of blackline. But unfortunately, the rootstocks that we use, whether it be black, Northern California black walnut, or paradox, these both actually will express a hypersensitive response because they're actually resistant to the virus. I know this is very confusing. I'm more than happy to discuss this further outside this meeting if you ever want to know more. But what happens is this actually creates a graft incompatibility. So you get this necrotic zone of dead tissue across the graft union because of the different reactions of the plant material, the way that I like to describe it, the two different genotypes, the two different plants that you have react differently to that virus, and the rootstock reacts in the sense that you get dead cells. So you get this graft incompatibility, cells die at the union, and then the tree on top is literally cut off from the ground and starts to die. Whereas your rootstock is doing great and it's pushing out suckers and it's driving you crazy, whereas the top of the tree is starting to croak. The best thing you can do about walnut blackline is when you find it, so certainly first call your farm advisor because you have a problem and we're happy to come out and check it out. Then we will look for a variety of different things that could be going on. And if we find this and we determine that it's present, it's pretty easy to diagnose once you find it. Then it would be in your best interest to remove the trees after harvest, basically before it spreads pollen again, because once that tree is infected, it's spreading infected pollen to trees nearby. So, and then you can kind of determine what you're going to do based on how old your trees are. Walnut blight is something definitely worth mentioning on a walnut disease talk. So this affects the fruit, stems, buds, and leaves, any green tissue, any growing tissue. The causal agent is also bacterium. So it's Xanthomonas I always struggle over this, arboricola pathovargoglandis. These are all different photo examples of what walnut blight might look like in the field. You can see it inside the nut there. You can see it on the stem, on the branch, on the catkins, on the flower, and then also on the fruit later on after that infection has matured. This is usually what I end up getting and sending off to the lab, unfortunately. It's a beautiful disease cycle for walnut blight here. Basically, you need to have water in order to have a problem. So early rains, early blooming varieties usually get stricken by walnut blight. The big catch here is when to apply, and I've had people ask me constantly, and I finally know the answer. So if you have a severe infection from the previous year, you will apply your bactericides during catkin elongation, whereas if you do not have a high inoculum level, you can just start applying at prayer stage with the female flowers. Again, this information is on UCIPM. Jim Atascavich is an absolute expert when it comes to this. He knows much more than I do about it, and I usually just say, let's leave it to him for further detailed questions. So another issue that you might come across in your orchard, you got dying trees and you pull them out of the ground and you come to find that their roots are just horrendously mangled around each other. So poor planting, will cause a tree to croak, but it'll probably take about 5 to 7 years because those roots have to grow large enough, thick enough to start choking themselves or each other out. So this is a really unfortunate reality when you're about 7 years in and your trees start to croak. So don't twist your roots. Make sure that when you dig the holes, you dig them big enough for the root system, and not try to crunch and squish that root system into a smaller hole. Don't twist the tree to get it to fit. Just set that tree in nice and gently and pile the soil back on top and pack it in nice and then take good care of it. Irrigation practices, I see this more often than almost anything else. We are finding with some delayed irrigation work and some water management that you'll hear much more about tomorrow and in great depth by Ken Shackle. We are finding that walnuts are probably, we're thinking that we want to irrigate them sooner than they may need it in the spring. And so we are working diligently on figuring out how to decipher when to start irrigating in the spring. Because by the time you get to July and August, it's too late and you're going to have to wait around another year to be able to kind of start working on fixing that problem. It's just you end up reducing the root system so much and it makes it very difficult for the tree to survive. So I do like to mention that, but again, tomorrow we're going to have a really great in-depth conversation or presentation about that topic. So please do tune in if you have a chance. It will be recorded and available on YouTube as well. So you can always come back and watch it later. Our malaria is definitely an issue. Admittedly, I don't see it very often, but it is out there. And this is like a perfect example of what we call a mycelial fan. This will cause your walnut tree to die over time, kind of a slow, sad death. And it can be very difficult to control. There really aren't too many options. Good sanitation, early detection, because you're going to want to remove it if you see it. Avoid ripping or disking in infected areas to reduce spread. Avoid overly wet soils. Do not introduce our malaria with equipment. Certainly certain rootstocks are more susceptible to others. And, the most susceptible English moderate is Northern California black, and extremely aggressive strains will even white about paradox. So really your best bet is to remove infected or dead trees as soon as possible, if unsalvageable. And I will leave it at that. I'll go ahead and open up the questions as well. I probably got through a little early, which is always a good thing. Let's see. All right.
Thank you, Kari.
Yes.
Thank you very much.
Great talk. Thank you. Okay, so identifying diseases of walnut and management strategies. So what is the causal agent for crown gall in walnuts? Is it Agrobacterium tumefaciens, Geosmithia morbida, or Cytospora chrysosperma? Okay, I'm going to go ahead and end the poll in 3, 2, 1. We'll share results. Yes, the answer is Agrobacterium tumefaciens. That is the causal agent for crown gall in walnuts. Fun fact, it's Agrobacterium vitis in grapevines. And we'll go to the next question. Okay, so you know what? I don't think I had this slide in there. So you guys get a little bit of a freebie here. Okay, so how can crown gall spread? So what do we know about crown gall? So it's spread by water. It's soil borne. We know that it's associated potentially to dirty gloves or soil. So what types of things would go into this? So if we're collecting paradox rootstock seeds from the ground and we're not using tarps, is that an opportunity for infection? If we're trimming the roots, if we're making a wound, so Agrobacterium tumefaciens really likes to infect wounds. So if we're trimming the roots or if we're pruning our suckers, if soil and water splash up into there, I think everyone got it. Good job. All of the above is the answer. Good job, everybody. So now we're going to be really careful about our pruners, right? Excellent job. Okay, do we have any questions out there in the ether?
Yes, we have one question, Kari. The question is about the upcoming frost. Will some of these upcoming frost events have an effect on walnuts after it being a dry winter and warm in the past week?
I think that's an excellent question. I think that, and I would really hope that some of my colleagues out there that are on the panelist section here would chime in if I miss something or if something needs to be changed. So I would say that walnuts are not emerging yet. We're still at the end of February, so they're still asleep. They're still dormant. I think that they are protected from the frost. That said, I think that other orchard crops, some of my peaches, some of my apricots are starting to bloom. So as those buds open up, there's an opportunity for freeze or frost damage. We do actually, I sent something out to our email list today, but we do have, Sac Valley Orchards actually has a really nice article on what to do about frost issues. It's more associated to almonds, and I think that the information is also valid for cherries and apricots, et cetera. In this case, you would be applying water basically to reduce the effect of frost, and you're only going to affect it to a certain height. So if we were to get a freak frost event in May, I'm not really sure to what extent the irrigation would help. Probably 10 or 12 feet, but I'm not sure if it's going to go up to 20 or 30 or 40 feet high. But with that said, it's still something that can be done. So I don't think that the walnuts are going to be affected by the frost, but I know Bruce Lampin is on the line and a few of my other colleagues might be able to chime in. Does anyone else have any thoughts on that? The effect of frost right now with walnuts? I think they're going to be okay. Going out on a limb here. Nobody else is saying anything, so I'm not sure if that.
There is actually in the chat, Luke chimed in as well as Bob. Bob is mentioning that he would not want the walnuts dry if the temperatures drop below or below 26.
Okay. Thank you, Mohammed. I didn't see the chat there. So yeah, so you wouldn't want the soil to be dry, and there is certainly, so Janine has seen winter kill only when the soil is dry. With no rain in the past 2 months, it's good to irrigate and have some soil moisture ahead of the freeze. However, please avoid standing water. When it freezes over, it prevents heat from radiating. Very good information. Some growers in the area with solid set irrigation plan to do active irrigation during the freeze in their coldest spots. And then of course, my question is how many people have access to water right now? I know here, I don't think we've filled the canals yet. So I think a lot of people are just hoping and praying that it works out. And so Janine says, agree with Bob, but to 28. Thanks, Luke, for posting my advice above. So yes, thank you everyone for putting in your additional information here. So there is likely value to having moisture out there against the frost damage. I'm just not really sure how many people have access to water right now. I think that's also another concern. And, maybe in the future we can try to change that. It takes time. Excellent question. Any other thoughts? So it is getting chilly. If you have access to water, you can certainly give it a shot. What I'll do is I will go ahead and post that Sac Valley Orchards article into the chat box so that you all have access to that link. I can pull it up right now. So I think that that concludes the session. I'm going to go ahead and post the link for the so it's an evaluation and it's DPR quiz and CCA credits. Everything is in that link. So I strongly encourage you to go ahead and follow that link, even if you don't need DPR credits, even if you don't need CCA credits. You don't have to answer the whole, you can just answer the first half. It's a pretty quick evaluation of the course, and it's also a pretty quick evaluation to get your DPR and CCA credits. So with that, I'm going to go ahead and look up that website for Sac Valley Orchards for frost mitigation, so that those of you who do have access to water can do something to try to help. Aside from that, I would say that concludes our program. So thank you very much, everyone, for attending. I'm going to stop recording, and I'm going to see if anyone's got their hands raised out there if they need my help. Thank you, everyone.